Okay guys, morning. Today we're going to chip away really a little bit more of assignment C, as much as we can do really without actually being in the classroom and doing the experiment. But again, the more we get a head start on this, the better. So the story so far, right? This is the assignment. Task 1 is you need to have done 3 different chromatograms. Now you haven't and you can't until we go back. So this is very much a next year job. But that's the first bit to do. And obviously we're kind of limited to what we can do until we do that, but there is stuff in the meantime. Task 2, you will have done by now, or at least you should have done by now, and if you haven't, get on it. Right, that was what we did the other day. There is a lesson video and all the rest of it attached to that, so make sure you have that done. Ideally as a table, right, because it's the easiest way to drop and drag to put it in, but make sure that's done if you haven't done it. Then we did task 3. Now task 3 you cannot do all of because you haven't calculated the RF values for each because you haven't done it, and you then can't identify it because you haven't done it. But you can do the third part, which is tailing. So talk about what tailing is, how we can prevent or reduce it. Again, that was the video from the other day, and so there's lots of information on that, and so again you can just drag and drop that in. But today we're moving on. So today we're going to look at task 4. Now this is the part of the evaluation. It's distinction level stuff, but to be honest guys, it's not that difficult. So you need to have done obviously the chromatogram to be able to do it. Part 1, 2 and 3 at the beginning you can't do anything about. That's evaluation. But the last part we can do. Look at your chromatogram for the separation of amino acids. Why don't all of the amino acids travel up the paper at the same rate? We're going to look at the structural formula of those 4 amino acids and include them in the report. What's the relationship between structure and RF value, and what significance is it that some side chains are polar and some are not? They have charges or whatever. So today what I want you guys to do is to find the 4 amino acids, which I'll talk about in a second, find their structural formula off the internet, find pictures and reference them. I haven't referenced them on here because you're going to have to go searching for them. But you need to find those 4 amino acid diagrams. We'll talk about the side chains, and we'll talk about what we're expecting to see and why, and then when you do see it, you can talk about it. Right, that's the plan. So that's what we're going to look at. That bit at the bottom is the bit we're going to do, because the rest of it you can't do till you've done the experiment. But the more we chip away at this, the less work you've got to do next year. Come in, we do the practical, we write it up, a little bit of evaluation, cut and paste your stuff over, done. Right, easy peasy lemon squeezy. So, I can't believe I said that, sorry. Right, what is an amino acid? Right, this is an amino acid here. Right, it's a very particular kind of molecule. At A-level chemistry we spent some time on it. In biology we spent some time on it. But as far as we're concerned in BTEC, it's just a molecule. But it's called an amino acid because it has 2 bits on it. It has an amine group, or an amino group, which is based on ammonia. Right, ammonia is NH3, so nitrogen with 2 hydrogens. It's basic. It's an alkali. Okay, so it reacts like a base. It accepts protons and stuff. But then the other end, we've got an acid group. So we've got a carboxylic acid there. Carboxylic acid's a C double bond O OH, and that's the sort of stuff that you'd find in ethanoic acid or vinegar or formic acid or methanoic acid, which is the stuff in ant bites and bee stings and those sort of things. Right, but they're amino acids. They've got an acid group and they've got an alkali group or a basic group, and that means they can do lots of quite exciting things. One of the exciting things is they bond together to make big molecules, which is basically how DNA forms and how proteins form. But the significant part is all of these amino acids are exactly the same, except for this bit in the middle. Right? You've got a CH, you've got the amino group at one end, you've got the acid group at the other, but you've got an R group down here. Now R just means alkyl, it means some sort of carbon-based group. The significance of the different amino acids is the R group changes. So all of the amino acids we're going to look at are just going to have a different R group on them. The rest is exactly the same. So what can those R groups be? Well, actually there's loads, there's tons. Now this, if you want to be really specific, okay, pedant mode on, this is what we call an alpha amino acids. There's different types, but as far as we're concerned, this is the only amino acid we're going to look at. And there's lots of them. Now we've got here the 20 common amino acids. This is a biology thing because your diet is important. When you eat proteins, you need to get amino acids into your body, and amino acids then can be built into proteins. They can be built into DNA. So it's really important. There are 20 what we call proteinogenic amino acids. Right? You don't need to know any of this for this, but just so you are aware, all of these have the same idea. There's the carboxylic acid group, there's the NH2 group, there's a side chain, the R group. There we go, little R group there. This is a big R group, there's another big R group. Look at that big R group there with a ring on it, another R group. And you can see some of these R groups are big and complicated, like this one. Some are very, very simple, like this one. So there's lots of different types. And it's the nature of the R group that makes it do different things, form different proteins, code for different proteins in terms of DNA. And there's 20 that are used in the human body. The key thing is the R group is different, right? The NH2 group and the C double bond OH group are exactly the same, just that R group, right? And that's important in terms of what we do in chromatography, because when you do the amino acids practical, and I'm going to ping the PDF attached to this so you can have a look through that practical so you get an idea of what you're going to be asked, because the questions at the end of that are exactly the same as the questions on the task. We're going to look at 4. We're going to look at arginine, leucine, glutamic acid and valine. And we'll warn you, I'm a chemist, which means I'm rubbish at pronouncing amino acids because they are biology. But let's have a look at the structure of them. There's arginine, there's leucine, there's glutamic acid, and there's valine. Now, I've deliberately found pictures that show the C double bond OH group and the NH2 group in the same position. So you can see here, acid group, basic group, R group. Acid group, basic group, R group. You see them drawn different ways, and I've deliberately picked ones that don't. Acid group, NH2 group, R group. Acid group, NH2 group, R group. Why? Because you can look at the R groups directly. Now, the key thing is here. Arginine has a basic side chain. It's got another amine on it. It's got an NH2 group. That means it's basic. There's a pole there. Now, think about poles. You've got the North Pole and the South Pole. You've got like a difference in magnetic charge. Now, you might remember way back in the dim distant looking at electronegativity. And this has got a difference in electronegativity because you've got nitrogens down there. So this is a polar molecule. It will have a sort of slightly positive end and a slightly negative end. Right? It's polar. Glutamic acid has got an acid group on it. It's got a second COOH group down here. And again, you've got very electronegative oxygens. So you've got a slightly negative end and a slightly positive end. What that basically means, what that means is you've got 2 polar molecules. Arginine and glutamic acid are both polar. Right, why is that important? Well, if you compare that to the other 2, these have got alkyl side groups. They've just got CHs on. Now, some of these are skeletal formulas, right? This is a skeletal formula, and this is a normal sort of molecular formula, or structural formula. This shows all of the carbons, as does this one. These zigzags, each zig and zag, that's a CH2, that's a CH2, that's a CH2, that's a CH, CH3, CH3. It's just skeletal formulas are just easier, but they do mean the same thing. What this is non-polar. It's just got carbons on, and carbon's not particularly polar. This has just got carbons on, so it's not particularly polar. So I have 2 polar amino acids and 2 non-polar amino acids. Right? That's crucial to answering these questions. Find the 4 structures, identify the 2 polar ones, identify the 2 non-polar ones. Why? Because there is an adage. There's 2 adages in terms of chemistry, in terms of solubility and attractions and things. Like dissolves like, and opposites attract. So, what does that mean? Well, I've got 2 non-polar molecules here. If I try to dissolve those in water, which is polar, they're going to struggle a little bit. Think about oil and water. Oil is non-polar, water is polar, and they do not mix. One will not dissolve in the other. Oil will always stay separate. Whereas these polar molecules will dissolve beautifully in water, perhaps less so in oil, because water is a polar solvent and they're polar molecules. So, like dissolves like. If you've got a polar solvent, it will dissolve the polar molecules. If you have a non-polar solvent, it will dissolve the non-polar molecules better. Now, there's going to be a certain amount of wiggle in this, but that's the first thing. Second thing is opposites attract. So if you've got something that likes to stick to something, chances are they're opposite. If polar molecules stick to the paper, that's because the paper is non-polar. If non-polar molecules stick to the paper, that's because the paper is polar. So, why are we talking about this? Because we're expecting, when you do this, you will almost certainly see, you will see actually, the two polar amino acids move to a similar extent to each other. The two polar molecules are going to go to roughly the same height as each other, and the two non-polar ones are going to move to the same height as each other as well, give or take. Now, why? Why am I going to get the two polar ones at one height and the two non-polar ones at the other? Or vice versa? Because of a combination of those two things. Like dissolves like and opposites attract. Because either the molecule is very strongly attracted to the mobile phase, or the solvent, because the mobile phase is polar and the molecules are polar, or the mobile phase is non-polar and the molecules are non-polar, or the molecule adsorbs, remember adsorption is sticking to the surface, adsorbs very strongly onto the stationary phase. So again, if the non-polar ones don't move very far, that's because the paper is polar. If the polar ones don't move very far, that's because the paper is non-polar. Or a combination of the two. A combination of the attraction to the solvent and the attraction to the stationary phase. Now, you won't be able to say which that is, because you just won't. It's a combination of things. But this is what you're expecting to see. Now, you won't be able to say which has gone higher till you've done it. But what you can do is find the four molecules, note the two polar ones, note the two non-polar ones, and talk about the fact that you're expecting the non-polar and the polar to move to the same height as each other because of this combination of two effects. Either the polar molecules dissolving the polar solvent or the non-polar molecules dissolving the non-polar solvent, or opposites attract. Polar sticks more strongly to non-polar stationary phase, or vice versa. So there's quite a bit to get your heads around. If you make a start on this, then again, it's just drop it in later. There's far less time spent doing this later on. So please do that for me now. Hang on to it. Save it somewhere where you won't lose it. Shove it in your OneDrive. It won't go anywhere. Okay, any problems, give me a shout. Otherwise, I'll see you all soon. Take care.